So now, after talking about independent assortment, let us try to understand what is the concept of linkage and recombination. Our next point is linkage and recombination. Now, to understand linkage and recombination, understand that in this particular scenario, both the genes are located on homologous chromosome, matlab same chromosome. Both the genes. In the first scenario, the gene A and gene B were located on two different chromosomes. There were 4 pens total over here. Now there are only 2 pens. That means that they are located on the same homologous chromosome. Now, if I ask you a question, how will you represent this kind of structure? Will I write like this itself, or is there any other method? The answer is we can write like this itself. So whenever you are writing like this, so the drawback of writing like this is you cannot know whether they are on the same chromosome. If they are on the same chromosome, you will be writing like this. If they are on the different chromosomes, still you will be writing like this. Anyway, let us come to the concept of linkage and recombination. So here, if you imagine, if these two are the chromosome and these two are the genes, now let us do the meiosis process. In meiosis, again there will be segregation. So when you segregate, how many total chromosomes are there in right now? So there are 2 chromosomes. So when gametes are produced, how many chromosomes should go in each gamete? The answer is half of 2 is 1. So now one gamete will receive this chromosome, another gamete will receive this chromosome. So this would be one gamete, this would be another gamete, right? So we are producing one gamete, capital A, capital B, that is this one, and another gamete, this one, small a, small b. Now the question comes, in this situation we were producing 4 types of gametes, but in this situation we are producing only 2 types of gametes. So what about the gamete called this one and this one? How they would be produced? How will the gamete capital A, small b, and the gamete small a, capital B, how these 2 types of gametes would be produced? The answer is they would be produced only and only if there is crossing over. If there is crossing over. Now what is crossing over? Crossing over means whenever these 2 homologous chromosomes were together in the somatic cell, some portion from this chromosome will get exchanged with the another homologous chromosome. So what is going, let us imagine this kind of situation. So let us say this chromosome, this part of the chromosome is exchanged with the next part of the chromosome. Now imagine this is called crossing over basically. So if this kind of crossing over occurs, in that scenario some cells will produce this kind of chromosomes, this kind of gametes, also this kind of combination, small a capital B and capital A small b. But what do you think? What are the chances of crossing over here? So in this scenario, the parental combination, these 2 are called parental combination, right? So parental where 50%, recombinant, these 2 are called recombinants. These 2 are called recombinants, these are called parental. So in this situation, what is the percentage of parental? 25 plus 25, 50%. And what is the percentage of recombinant? Again, 50%. I am talking about independent assortment. In that scenario, it would be 50-50. But what about this case? In this situation, majority of the cell will produce these kind of gametes. Only few, whenever crossing over occurs, crossing over is a chance phenomena. So the number of these 2 types of gametes, that is recombinants, would be much lesser. Let us try to understand with an example. Let us imagine that these 2 are produced, let's say 40% and 40%. Then what would be the percentage of recombinants over here? 10% and 10%. Now question comes, how many recombinants will be produced? It depends on which thing? So it depends upon the distance. Now let us imagine if the distance is lesser. Let us imagine if the distance between the genes would be less. So the chances of crossing over would decrease, right? Recombinants are produced only and only when there is crossing over. Okay, recombinants are produced when there is crossing over. Crossing over depends upon distance. If there is higher distance, crossing over would be more. If there is less distance, crossing over would be less. So now I have decreased the distance. So as I have decreased the distance, again the number of recombinants would reduce further. Let us say it would become, let's say 5%, 5%. And this would become 45%, 45%. So on what factor does it depend? How many recombinants would be produced? So it depends on the distance between the genes. So now here, whenever genes are together, like capital A, capital B will be going more together as compared to capital A, small b. So this concept is called linkage. The genes are physically linked, they are closer. So that concept is called linkage, and these two, whenever recombinants are produced, that is called recombination or recombinants. So here, these two represent linkage while these two represent the recombination. And remember, higher the distance, higher crossing over. Higher crossing over is equal to more recombinants. And less crossing over means less recombinants. That's it. Thank you.